Thank you for the kind introduction, and I'm happy to have such a great opportunity. Today, I'm talking about the genome-based machine learning model for zoonotic viral prediction. So as you may know, many infectious diseases have been caused by animal-derived viruses, and previous study suggested hundreds of thousands of viruses hidden in animals were estimated to have human infectious potential. So, and therefore, monitoring of animal viruses, not in human viruses, not only human viruses, is needed for the future pandemic preparedness. However, the problem is that investigating viruses in animals, wide range of animals, is labor intensive and time consuming. Therefore, we have been working on the digital transformation for supporting biological research. For example, we are trying to conduct the viral infection surveillance in animals by reusing public sequencing data or constructing the machine learning model for genotype to phenotype prediction. So today I'm talking about the second topic. Metagenomic analysis have rapidly elucidated viral genetic diversity in animals. However, analyzing viral phenotypes such as infectivity or pathogenicity still requires substantial human effort. And in particular, human infectivity, it is very important phenotype to relevant the zoonotic infectious diseases have still unelucidated for most of animal viruses. And therefore, in this study, we aimed to develop the machine learning models for predicting viral human infectivity only using viral sequences as input. So first, to construct comprehensive dataset for viral infectivity prediction, here we collected the viral nucleotide sequences and their host animal labels from the NCBI viral dataset and provided 29 times more data than previously available, which covered 26 very wide range of viral families. And next, to construct the genome-based model for detecting viral human infectivity, we conducted the fine-tuning of nucleotide language models. So as you may know, the BAT models can pre-train the context-like general rules from the large number of nucleotide sequences. And such a fundamental model can be used to construct the task-specific models by fine-tuning with the limited labeled data. Following this strategy, so we constructed a new model, BAT-infect. From here, we move on to the results section. So this panel shows the PRAUC score of our BAT-infect model and existing models, human virus finder, APBJC, and Zoonotic rank. So as shown here, we found BAT-infect models outperformed existing models across 18 viral families. And also we found the improved performance of our models on segmented RNA viruses, which have been neglected in viral infectivity prediction tasks owing to the limited data availability, despite containing these viral families contains the very high-risk zoonotic viruses. So these results indicate that our approach is the fine-tuning the nucleotide language model with the large-scale curated dataset substantially boosted the BAT-infect model performance. Furthermore, we evaluated model applicability for shorter viral sequences. So for detecting human infecting viruses from high-throughput sequencing data, such as the short-read sequence data or the various lengths of context sequences. As a result, we found that BAT-infect and DPAC-BIA models maintained consistent performance for most viral families, regardless of the input length. So these results indicate that only specific models are suitable for mining human infecting viruses from high-throughput sequencing data. And next, we also checked the model performance for newly identified viruses. To check this, so we trained these models with the data identified up to the 2018 and test their performance in predicting using the future viral dataset. As a result, we observed comparable median F1 score in most models, except for the Deepak beer, suggesting a high level of model predictive capability towards novel viruses. Based on the results so far, you may consider that we have successfully constructed the model with good performance. However, our high-resolution evaluation based on the phylogenetic analysis identified difficult to predict viral lineages for all current models. So in this panel, we mapped the phylogenetic relationship of viruses alongside their prediction probability. The upper panel shows the phylogenetic tree of viruses, and the middle panel shows the viral properties such as the viral genus or host levels, and the bottom panel shows the predictive probability generated by the distinct 5 models. So as shown here, so please focus on the green one, so hepatovirus. So in this, the genus, so human infecting and animal infecting viruses phylogenetically are distinguished, which seems to be relatively easy to predictable. And indeed, all models showed a high predictive performance, around the 0.8 F1 score. On the other hand, the model performance dropped in the genus Flavivirus, which was characterized by frequent change in infectivity during their evolution. These results suggest that insufficient predictive capability of current model for viral lineage, specific viral lineages where the zoonotic infectious potential has been acquired repeatedly. Furthermore, we also revealed another limitation regarding the SARS-CoV-2 related viruses. In this slide, we show the ranking of the predictive probability and with each column corresponding to each models. And the upper and middle panel show the result of the time split dataset, and the bottom panel show the SARS-CoV-2 related viruses. As shown here, so we found that no models accurately identify the human infectious risk of SARS-CoV-2 related viruses, despite all models achieving high predictive performance for time split dataset. So finally, let me summarize the key points of our research. So in this study, we aimed to improve the viral infectivity prediction model by creating dataset covering various viral families and developing new models based on the nucleotide language model. And these approaches boosted the predictive performance on the bat-infected model. Furthermore, these models trained on data up to 2018 showed high predictive capability for viruses emerging afterward. However, we also found the general limitation in all current models, the difficulty in alerting the human infectious risk in specific zoonotic viral lineages, including SARS-CoV-2. So in other words, our taxonomic-based validation highlighted difficult to predict viral lineages. And these viral lineages have been overlooked due to the uniform model evaluation for category viruses. And these findings contribute to emphasizing an essential area for model improvement and stringent future pandemic preparedness. So thank you for the listening.
Thank you for your wonderful talk. Sorry, now behind schedule. Please, only one question. Anyone? No? So I have one question. How will you deal with the lack of comprehensive data sets in the future?
Yeah, it is very important point and struggle that point. But actually, maybe it is impossible to experimentally validate the infectivity, but I think the data imputation based on the solid dataset, so I think it is possible the way to the solution.
Okay, I understand.